Well, I want to talk today about some ideas that were inherent in my title. I hope you can see all right what I wrote over there, because I'll talk from this blackboard here, but I put two things that I learned or was told when I was in Rochester in the 1960s. One of them, the first one is by Dick Lewontin, and if you can't read it from there, it says, genetics went downhill when geneticists became interested in Drosophila. Now, Dick was a Drosophila geneticist. What he meant by that was that Drosophila had been extremely useful for studying questions about genetics, about replication, recombination, etc. When it got to the point that they sort of ran out of easy things like that to do, they focused on the organism itself, and that was a problem. John Roth actually has written very eloquently articulated pretty much the same principle for bacterial genetics, that when bacterial geneticists really become interested in bacteria, or when phage geneticists really become interested in phage rather than what you can learn from them in general, of general things, why you're at a bad stage. The second one came from Max Delbruck, and it says if a question is of no general interest, the further effort should not be, if a question is of no general interest and has been given a sufficient explanation, it should not be studied further. Now, I must confess, I never heard Delbruck say this. I was told this by Luria, who said once that he had rejected a paper to virology on the grounds that he told the author, well, look, you're the only person in the world who's interested in this question. You've already published one paper, so that gives you a literature to read. And so there's no point in publishing anything more. I mean, the key word here is sufficient. A sufficient explanation is not necessarily the right explanation, but getting everything right about the classical organisms isn't necessarily a particularly worthy enterprise. And I'll come back to that a little later. So then, the third thing that I have there, when I started studying Lambda, I was studying primarily Lambda integration and the mechanism of formation of specialized transducing phages, but one of the most interesting questions about Lambda was that it's a temperate phage. It can choose either lysis or lysogeny as an alternative, and the question is, how does it decide which one to choose? And there we have what I've referred to as the Gospel according to St. Mark. That's Mark Potashny, who wrote a book called A Genetic Switch, which he later has said might be modified to be called an epigenetic switch. There's no, because it's a switch which involves no change in DNA sequence, simply two alternative states of the same DNA sequence. And this was described in detail in Mark's book. The essential features of it are here. This was based on biochemical experiments that Mark had done and a lot of previous genetic work that had been done by Dale Kaiser. And what it said was this: there is, in a lambda prophage, there is a section here which is about 1 kb in length. And if you just take that section and nothing else, it has two stable states. It can either be transcribing the C1 gene and making more repressor, or it can be transcribing the CRO gene and making CRO, and these things end up being mutually incompatible. You end up with a competition actually for one of these sites here. These are the leftward operator, the rightward operator. Each one of them has 3 oligonucleotide length sequences similar but identical to each other, which bind both repressor and CRO. And the key thing ends up being a battle for the occupancy of OL3. Whichever one occupies OL3, repressor or CRO, wins. And that is because of how it affects this transcription. You can make repressor from here, and repressor can, at a very low level, and repressor can turn that on to a very high level. So if you have a high level of repressor, you stay with repressor, you remain repressed. If you have a low concentration, and if CRO occupies this, then it prevents repressor from turning itself on. And then you go this way. And in the whole phage, if you go this way, you go toward lysis, because this is transcribing the late genes. And if you go this way, you establish lysogeny. So the battle is here. The battle is between CRO and C1. So that's a sufficient explanation under Delbruck's rule for how it works. It's in all the textbooks pretty much, and Mark described it very well in his book. I don't follow Delbruck's statement here completely because of the first part. If a question is of no general interest, to me this question is of extreme general interest. It's not a detail about the biology of Lambda. It's at the heart of things. Is this really the point where the decision is made? And the thing there is that there is an alternative to it. And that has been, well, I will attribute it to 4 people: myself, Shankar Adya, Don Court, and the late Amos Oppenheim. The only reason I put myself first is not because I have done most of the work. I have done very little of it. These other people are responsible for most of the experimental work that goes into this, but because this is what I think, and I'm not sure that all of these other people think exactly the same thing. This one feature here that's left out of here, and Mark knew he was leaving it out in this discussion, is that repressor can be made from another promoter that's over here. And this is within the gene, the C2 gene. I hope that color shows up well because apparently none of my colors did. And within that gene, there's a promoter called PRE. Does that show well?
No.
No. Okay, well, let me try it. Should I try the yellow one?
That ain't better?
No. Okay, well, anyway.
Nope.
Okay, well,
I think we're back to white.
maybe I better just do it in white then. Okay, so that's PRE. PRE, and it's within the gene C2, and C2 turns on this promoter, and this promoter transcribes C1. And so this is the way repression is established in a cell. This was Dale Kaiser's work that showed this, and you had to have such a repressor because this transcription is very weak. If you just relied on spontaneous transcription from PRM without having C1 to turn this on, then you would very seldom make repressor. And instead, you want to be able to make it rather frequently, so you have to have another promoter here to give you some C1 to get things going. And so Dale had shown this clearly. The question has been, does C2 just serve some kind of ancillary function where you put a little repressor on the table and then repressor and pro are able to duke it out for possession of this side? And that's what has been implied by a lot of studies, including some from another lab at Stanford, which which could reproduce reasonably well the data on the effect of multiplicity on the system by assuming a lot of parameters, not all of which were measured, but which ends up really, if you examine what is said, as being, again, that this is the decision point. Now, what this hypothesis says is that this is not the decision point. That the decision point is over here at this promoter, and that the antagonists are pro and C2. And when you get to the point of determining possession here, you're already in a cell that's committed to lysis. And the facts here that are relevant are that the Cro protein is relatively stable, so it accumulates with time. Whereas C2 is unstable, even though the cell does its best to stabilize its sum. So now what does that mean? If you have, Cro can turn this operator off. So it turns the rate of transcription in this direction down, and it keeps turning it down further and further. If you have an unstable protein, you have to have a high rate of transcription in order to build up a high concentration, because crudely speaking, if you have a half-life of 2 minutes, you are looking at what accumulated in the last 2 minutes. So the argument is that you accumulate Cro, and once the Cro gets beyond a certain level, then you're no longer able to make enough C2 to turn this promoter on, and then you are trapped in the I guess it's in the lytic phase, because the Cro has given you the clock that the accumulating Cro has turned things off. So now, why do I like this idea? The main reason is because if you look at the Lambda situation, the entire situation with Lambda, C2 actually acts at 3 places. It acts in turning on the integrase gene, which is here. the C1 gene, which is here. And then it also acts here to turn on a promoter that I think is sometimes called pre-anti-Q. It is a message in the wrong direction for the late proteins. So this is turning late protein synthesis off. And all of these syntheses are controlled by C2 binding to a promoter which has in its minus or around its minus 35 region this sequence, TTGC followed by 6 bases followed by TCGC again. This is in double-stranded DNA. And now, the point of this is that it doesn't make much sense biologically to either have, unless you coordinate these things, so that they're either all off or all on. If you do anything else, if you integrate the phage but don't make any repressor, that's a waste. If you turn off the late functions but don't repress, that's a waste. The only way to go cleanly from one system to another, situation to another, is to either have them all turned on or all turned off. And therefore, C2 has to be in control. And the only way you can really put C2 into control is by saying that C2 is controlling this synthesis as well as these. And so the competition has to be between C2 and Crow. So that's what we think. I've tried to persuade, well, I've tried to persuade some young people to study this more, but not really hard because it's not a good problem for a young person to study. Young people want to make their reputation, and the general idea is, well, this is in the textbooks, it's established. Who cares if it's not quite right? But I think it's really important to get this point straight. It's one that is of sufficient interest, it is of big general interest. So what we have been studying recently, and this is my lab, which mostly consists of Alice Campbell and myself at this point, is this control of integrase. And we've been studying in Lambda as compared to a Phage 21, which is a Lambda relative. Now, the thing about Phage 21 is it doesn't have a promoter of this kind in the right position to be turning on the integrase gene. What it does have instead is downstream from integrase, beyond the attachment site where the phage integrates, about 160 bases further on, there's a site called SIB. This was discovered by Gabriel Guarneros some time ago, that this site can control from downstream the synthesis of integrase. I won't go into the details of how that happens, but basically you have a double-stranded DNA here, and if you transcribe it from the PL promoter over here, you go through that and make a complete stem and loop, and the stem is then cut by RNase 3 and other RNases digest backwards into the integrase message and turn the integrase gene off. If you're transcribing from the integrase promoter, you stop about here, and you get something that is a stable message that isn't subject to that kind of degradation. But the thing that is tantalizing about Phage 21 is that if you look at this sequence in its double-stranded DNA stage, it has this sequence represented several times. That is, this has another PDGC6 next to it. In some of these cases, there's one base is off, but that's known to be the case in some of the phages where C2 works anyway. And the other thing is that in some cases that you have this configuration. Which gives you also a C2 recognition site in the other direction, TTGC, 6 bases, TTGC. So we find this tantalizing. We think it means that somehow this is controlled by C2 in page 21, is page 21, but it's controlled by a different mechanism than it is in the case of Lambda. In the case of Lambda, you have a promoter here. In this case, you have a downstream effect. I won't go into what the possibilities are for that. But anyway, that's what I'm spending my retirement years trying to sort out. And I feel this is one appropriate thing for superannuated scientists to be doing. And so if we have real luck on this, you'll hear from us eventually about it. Thank you. Yeah.
So Alan, the Lambda switch is pretty complicated if you compare it to some of the others. I get the feeling that it's on the rather complicated end of the spectrum. Why do you think that might be? And have you compared the genetic switches of other phages with this particular one?
No, I don't think so. And I don't know quite which pages you're talking about at this moment, actually.
If you look at environmental phages, for example, many of them seem to have a repressor akin to C1 and not much else.
And how does it give you a bistable switch?
It may not be.
I see.
The bistable switch may not actually be that common, I guess.
Well, maybe. Well, I guess I should emphasize this Potashny picture is in all the textbooks, and I think rightly so. I mean, if you're just looking for a model of a bistable switch that you think might explain what happens in the mouse brain or something, well, this is fine. This works. It is there. It works. We are still hung up on the question of, is it the way Lambda does it? And I think that's important, despite what Delbruck said there, because I think that the question is really of general interest. It's not one trivial aspect of Lambda biology. Doug.
So you think that in page 21, do you think that int is constitutive, or does it get turned on? You have working hypothesis, I guess.
No, I think, well, we've talked a lot about how the C2 here could be affecting the integrates. I mean, the prediction of this would be that if you have C2 around, it would do something to stabilize the synthesis of int. So I don't know whether, I don't expect it would be constitutive at the same level, at least, with or without the C2 being present. But anyway, we have no facts there.
I guess, sorry. So Delbruck, I guess, was very categorical.
Yes.
And kind of sure that he knew where science was going. But, I mean, basic research, isn't it this fantastic discovery of things that you couldn't predict beforehand that is part of the magic of what we all do?
Yeah, sure.
And that just, I mean,
But I think what Delbruck is saying there is that, I mean, certainly you make accidental discoveries, but the idea that if you're going to just wander into a lab and do a random experiment, that you're going to make an important discovery, is less than if your experiment is targeted towards something important, and then, of course, it may come out that it doesn't answer the question you asked, but it may answer some more interesting question instead.
So I just want to comment that at least when I first started going to Cold Spring Harbor conferences in the early 70s, the rule was that if you couldn't explain it on a board like that, then it wasn't worth talking about. Still very good at that time. Thank you very much.